Welcome to Pharmacology World. In this session, and in the sessions that follow, I'm going to guide you through the world of pharmacology, focusing on the key, high-yield aspects of pharmacology. The idea is that these videos are short in length, but long on explanation. A key goal of mine is to help you understand the mechanism of action of drugs, and when possible, describe how this mechanism of action relates to and explains the therapeutic uses for that drug and the adverse events that are seen. So whether you are a medical student, a pharmacy student, nursing, PA, or a foreign medical graduate, I believe these sessions will help you fill in the gaps from class, as well as a means of prepping for standardized board exams. In many of the sessions, I'll put quotes at the bottom. Sometimes these are quotes from famous individuals that figure prominently into medicine. Others are quotes that I've made up that hopefully will be memorable enough to help you remember the class of drugs and their mechanism of action. The quote that I put here is from Sir William Osler, and it reads, "One of the features which is thought to distinguish man from other animals is his desire to take medicines." So one of the first things we need to do is understand the key terms that are used in pharmacology. First off, what is pharmacology? Pharmacology is the study of the science which underlies the interaction of drugs with living systems. In other words, it's the basic science that helps explain why drugs do what they do, cause the adverse events that they cause, and have drug interactions. Within pharmacology, there are two subdomains: pharmacokinetics and pharmacodynamics. Pharmacokinetics deals with the effects of the body on the drug, and you see the acronym ADME or ADME. ADME stands for absorption, distribution, metabolism, and excretion. These are all the processes that are in place that mitigate whether a drug is going to be effective or not, and how long it's going to persist in the body. The other subdomain of pharmacology is pharmacodynamics. This deals with the effect of a drug on the body at a molecular level. In other words, how does a drug actually interact with its target? Is it binding to a receptor, to an enzyme, to a certain population of cells? It's really the molecular details of how a drug works. Then we have pharmacogenetics. That's another term that we should be familiar with. Pharmacogenetics deals with how genetic alterations can affect drug action. We'll see this come into play when we talk about metabolizing enzymes called cytochrome P450s, which are different between individuals. So there are genetic differences between individuals that guide how they metabolize drugs and how they react to certain types of drugs. Then we have the term therapeutics. Therapeutics is often erroneously used interchangeably with pharmacology, but it's actually a distinct area. Therapeutics is the clinical use of drugs to diagnose, prevent, or treat disease, or prevent pregnancy. When I think of therapeutics, I think about clinicians in a hospital setting or in a clinic using these drugs with patients, whereas pharmacology is the basic understanding of how and why the drugs work. Next we have pharmacy. Pharmacy is not the same thing as pharmacology. Pharmacy refers to the preparation, compounding, and dispensing of chemicals for medicinal use. Toxicology deals with the undesirable effects of drugs on living systems. And then we have some more granular terms that we'll come back to in later sessions. Effective dose 50 or ED50. This is the dose at which 50% of patients show a defined response. And then we have efficacy and potency. From a pharmacologic perspective, efficacy and potency have very defined meanings. Efficacy refers to the ability of a drug to produce a maximal response, whereas potency refers to the amount of drug necessary to elicit a desired response. And again, we'll come back to these terms later and see what this means in terms of drug action. Finally, we have tolerance. Tolerance describes the diminished physiologic response to a drug after repeated administration of a constant dose. I'm going to start off by taking a few minutes to talk about the history of drug legislation and drug regulation in the United States. I think it's worthwhile to understand a little bit of history behind pharmacology. The first act that I want to talk about came about in 1906. It was the Pure Food and Drug Act. This act was the first federal legislation that had to do with drug safety and efficacy, and it required accurate labeling of ingredients, and it sought to prevent adulteration of products with toxic or inactive ingredients. This legislation came about because of patent medicines that were popular in the early 1900s. There were wild claims that these patent medicines could treat a variety of conditions, but they often contained addictive ingredients such as morphine and cocaine, which will obviously make a sick or depressed patient feel much better. Following the Pure Food and Drug Act of 1906 was the Food, Drug, and Cosmetic Act of 1938. This act required evidence of drug safety before marketing, which is a marked improvement over the Pure Food and Drug Act. It established the Federal Drug Administration to enforce this act, and it gave legal authority to the USP, or the United States Pharmacopoeia, to govern composition of all drugs marketed in the United States, and it established standards for drug labels. This act arose in response to greater than 100 deaths that occurred in 1937 due to contamination of a sulfanilamide elixir with diethylene glycol, which is chemically related to antifreeze. The next legislation I want to mention are amendments to the Food, Drug, and Cosmetic Act. The first is the Durham-Humphrey Amendment of 1952. This created a legal distinction between prescription and non-prescription drugs. Then there was the Kefauver-Harris Amendment of 1962. This required demonstration of drug safety and efficacy in animals and humans prior to marketing. So this is a refinement of the idea that drug companies need to provide evidence of a drug's efficacy before marketing it. Now with this act, they have to demonstrate that the drug is safe and efficacious in animals and humans. The Kefauver-Harris Amendment arose in the 1960s in response to the thalidomide disaster. Thalidomide was developed by a German company to combat symptoms associated with morning sickness in pregnant women, although it turned out that thalidomide interfered with limb development and resulted in massive birth defects. Finally, we have the Orphan Drug Amendment of 1983, which through tax incentives and marketing exclusivity sought to encourage drug development of agents for rare diseases. And by rare, we mean fewer than 200,000 people. Then there was the Drug Price Competition and Patent Restoration Act of 1984, which accelerated approval time for generic drugs, which are demonstrated to be bioequivalent. And then finally we have the accelerated drug approval for new drugs to treat life-threatening conditions in 1992. This amendment was driven by the AIDS and cancer epidemics, and it allows patients with these diseases to be treated with investigational drugs prior to completion of clinical trials. Before a drug can be marketed and given to a patient, it needs to progress through a series of studies that evaluate its efficacy and potency, and importantly, its safety. The drug approval process entails preclinical studies, phase 1, 2, and 3 clinical studies, and phase 4 post-marketing surveillance. The goal of preclinical studies is to establish a pharmacologic profile for the agent under investigation. This is accomplished by using a variety of methods, such as in vitro studies with isolated cells and organs, receptor binding studies to understand the receptor-drug binding characteristics, in vivo studies using animals or animal models of human disease, and this brings up an important point, is that drugs are lacking where a good animal model of the human disease does not exist. Preclinical studies also assess the potential therapeutic use. Many drugs have started off targeting one disease, but ended up being marketed for a completely different disease. The preclinical studies help determine the pharmacokinetic profile for the drug and the toxicological effects. Preclinical studies help identify metabolites, provide evidence of bioavailability, that is, how much of the drug that's being administered actually gets to the site of action, and it helps establish the principal route of administration and the rate of elimination. In regards to toxicological effects, preclinical studies shed light on the potential hazards for a drug and allow investigators to begin to predict that hazard or that toxicological effect and its likelihood of occurring in humans at therapeutic doses. An additional focus of preclinical studies are to assess the mutagenic, carcinogenic, or teratogenic potential for a new agent. Mutagenesis entails looking at any potential effects on DNA that might be linked to carcinogenicity or teratogenicity. Carcinogenesis, on the other hand, the goal is to determine if repeated chronic doses of the agent under investigation can cause tumor formation. And finally, new agents are assessed for their potential to be teratogenic. That is, do repeated doses affect fertility, implantation, fetal growth, generation of fetal abnormalities, or neonatal growth. In between preclinical studies and phase 1 is the investigational new drug application. I don't want to belabor the point, but at this stage, the data that's generated in the preclinical studies can be submitted to the FDA in the form of an investigational new drug application. This application has information on the composition and source of the drug, manufacturing information, the data from the animal studies, clinical plans and protocols, and names and credentials of physicians conducting the trials. The goal of phase 1 studies is to determine the pharmacokinetic parameters of the drug in healthy volunteers. This is a small population of individuals who do not have the disease of interest. It's carried out by pharmaceutical companies or major hospitals. In some cases, the patients with the disease in question may be enrolled. Toxicity is evaluated, and routine hematology and biochemical monitoring of liver and renal function is also assessed. The results of pharmacokinetic and toxicological analysis serve as the basis for determining the doses for the subsequent phase of clinical trials. The patients involved in these studies are provided with informed consent to allow them to understand why the drug is being administered and what the potential adverse events are. The goal of phase 2 studies is to determine the pharmacology of the drug in the patients with the intended clinical condition. This is still a small group, but larger than phase 1. The principal aim is to define the relationship between the dose and the pharmacologic and/or therapeutic response in a small number of patients. Phase 2 studies may be divided into smaller subphases. The goal of Phase 3 studies are to compare the safety and efficacy of the investigational new drug with that of another substance or treatment approach. They often employ a larger group of patients with the disease at multiple sites across the country. Different study designs are used. Random placebo-controlled studies involve randomization of patient populations, and sometimes there is double blinding of the studies. Phase 3 studies may also involve what are known as crossover studies. Crossover studies are done in cases where it is unethical to administer placebo only. In these cases, patients are switched between placebo and treatment, either experimental or established treatments. Phase 3 studies look at what are called primary and secondary clinical endpoints. Primary endpoints are dichotomous outcomes. For example, the patient did or did not survive, did or did not improve in capacity to function. Secondary clinical endpoints are what are known as continuous outcomes. For example, decreased levels of plasma glucose or changes in blood pressure. If the Phase 3 clinical studies are successful and they're approved, then the drug manufacturer can apply for what's known as NDA status, or New Drug Application status. Finally, there are Phase 4 studies. This is also known as post-marketing surveillance, and the goal of the Phase 4 study is to monitor drug safety in the general patient population following FDA approval. This involves ongoing monitoring of drug safety under actual conditions and is sometimes referred to as pharmacovigilance. Pharmacists and physicians play an important role in this process by reporting adverse drug events, and there is no fixed duration. A recent example of a drug that was pulled from the market based on Phase 4 post-marketing surveillance is Avandia, also known as rosiglitazone, which is a drug that's used for type 2 diabetes. Finally, we get to drug naming nomenclature. It's worthwhile to understand that drugs have many different names: a chemical name, a generic name, or a brand name. We'll be using the generic name for the drugs that we discuss in all of the Pharmacology World sessions.